started.
As long as you don't make me too, as long as you don't make me too red. Should I share my slides or?
I think, sure. In the meantime, you might want to share your slides. Hi, everyone. Thank you for participating today in the Spatial Omics Seminar Series 7. And today we are very excited to have Professor Dana-Pierre. And briefly, Professor Dana-Pierre received her PhD from Hebrew University in computational biology. And after that, she did a postdoc in less than 3 years at Harvard University. Then she joined as a professor to Columbia University. And then there she stayed about 16 years. And then recently, she has been leading the computational and systems biology program at the Memorial Sloan Kettering Cancer Center. And she also has recently joined HHMI. I think that's a really, really good news. And that she's been leading the efforts in her lab at MSK to combine single-cell technologies, genomic data sets, and machine learning algorithms to address fundamental questions in biomedical science in general. And we were just talking about a few technologies before with Dr. Pierre, and today we're excited to learn about the new developments in her lab to impact spatial omics biology and beyond. And without further ado, I'd like to turn over to Dr. Pierre.
Thank you very much for the intro. Just for all you trainees out there, I just want to, you said, wow, I did a postdoc in 3 years. And I would like to say that it's highly unrecommended. While I managed to land a faculty job, I was not ready for faculty when I started because I didn't cook enough in my postdoc. So the first few years of faculty was very chaotic. And if there's any trainee out there, this is not a race. And I encourage you to use your postdoc to cook, to think deeply, to develop a deep plan so that when you hit faculty, which is 1,000,000 times more stressful and harder than a postdoc, you are well mature and ready to hit the ground running rather than walk around aimlessly and succeed by sheer dumb luck. Back to the topic and away from, advice. When I started at MSK, we also initiated the Human Cell Atlas project, which is now almost 6 years old. And the goal was really to get at in vivo biology, to try and understand humans and beyond humans, even mice. A lot of experiments were done in vitro, and now with all these single cell technologies, what this enables is to really profile at depth in real tissues in vivo, even in humans. Now, we've done really good about gathering the parts. There's over 2 million lung cells in the lung atlas. We have over 10 million immune cells. There are many additional atlases that are above the million. And I think that within a year, we'll be at 10 million cells for our major organs. So we're really, really good at collecting the parts. But one of the things that I've learned over the past few years is that you really need to understand how these parts are put together. These parts are put together in tissues. These tissues have structure, they have organization and architecture in which the cells work together to do something in concert. And actually in disease, it's often this architecture that goes wrong more than any individual part. And so we really need to be able to push the envelope of how to understand tissue biology. And up until recently, most of our viewpoint into tissues were H&Es. Now we're at a time where finally technologies are really coming on board. There's a rapid proliferation. This actually happened frustratingly slow. I was very excited about spatial technologies 7 years ago. And the tech dev here was harder and slower. And all you people out there in tech dev that are developing these technologies, kudos on the perseverance, and there's going to be an explosion of new technologies coming out very shortly. But what's really lacking is the computation. Anything from basic image processing, cell segmentation, even image alignment through data integration. It's a big thing to try and take these modalities that sometimes can only give you 30 or 100 markers or molecules per image, and really integrate it with all the other modalities that we're collecting, let alone, beginning to interpret and saying, well, what does it all mean and how can we learn biology from it? And new data always requires new algorithms, and out of the box ML algorithms is something my lab has really learned. And now I'm sort of quoting Doron from my lab, who is the first story I'm going to tell. So I'm going to tell you 3 stories of some methods that we've been developing in the lab to try and deal with these modalities. Here we're looking at IBEX, but this is general for any wide field imaging. At the beginning, I was working a lot with MIBI and similar data, site-off imaging, and these give you really, really little tiny windows. Also, the Visium gives you tiny windows. And here's an entire lymph node, and there is so much variation and so much structure in an entire organ. So being able to see a wide view is very powerful. Now, when you zoom out, these pictures look very, very pretty. This is another picture of a mouse lymph node, and it looks like we have a really good quality image of the entire field. Things begin to get wrong when you zoom in, and this is after perfect linear alignment. So this is the most optimal linear alignment possible, and what you're seeing here is individual cells, and I'm basically plotting the same membrane marker, CD45, and you see that the membranes are not aligned. These are supposed to be perfectly aligned. Not only are they not perfectly aligned, but each of these little views have a different degree of distortion. There's no single linear transformation that can really align these membranes together to give perfectly aligned images. And we see this in pretty much every wide field technology that we've looked at. The one with the most abundant data is CISA from Peter Sorger. So Doron developed a new algorithm, and it's a new type of autoencoder. So most autoencoders or most neural networks, they actually try and learn a new value for the pixel. So basically it's focused on a pixel and you want to change the value of that pixel. Here, instead, we decided to learn the distortion directly and to take a pixel and figure out where does it need to go? How's it been distorted? Where is its new pixel location? And for this, we learn a function that pretty much sends the pixel to where it needs to go. And we learned this in an unsupervised manner using the fact that we have in each of our images membrane markers, particularly for immune systems. Not only is there CD45, there's CD3. We have a lot of membrane markers that we can use, and we can actually tell the autoencoder, I want you to find me the transformation that will best align the membrane from one iteration to the reference. And this actually gives us a first of its kind neural network that can really learn local nonlinear mapping to remedy distortion between rounds. So pre-alignment, we saw it was pretty crappy. Here we use the state-of-the-art nonlinear alignment algorithm. This thing took hours to run, and it still doesn't cut it. And here is Mirage, and as you could see, it perfectly aligns. And this is not a cherry-picked example. You pretty much can take any 2 iterations and any 2 fields of view, and the alignment, it's very hard to find an area where the alignment doesn't work with Mirage. Now that we have Mirage, we can actually really be confident for each cell, what exactly is that cell expressing, and look at entire tissue and look at different regions across the tissue. For example, in the lymph node, the germinal center and the T cell follicle. And we can zoom out and see the whole picture of the tissue and zoom in at increasing resolution inside the germinal center. And inside further, we have full single cell resolution, full confidence of what each of these single cell is expressing, and a whole range of color in our 30+ marker panel to look at what each of these individual immune cells in the lymph node is doing, thanks to our very accurate alignment, which is soon to be available. Switching gears, after doing low-level processing, we want to figure out what we can do more, and particularly data integration and data interpretation. This is a joint work with the lab of Elaine Fuchs. It's been a really fun collaboration, recently accepted at Cell Stem Cell. It was really done by a trio of gifted powerhouse experimentalists that collected some of the most amazing data that we've had a pleasure of working with. And all the computation and analysis was spearheaded by Tini, a postdoc in my lab. So we really wanted to understand the gut and the gut stem cell niche. And there's both the small intestine where the gut is built of villi and crypts, as well as a large intestine. And while I think the gut, the intestine is pretty much the most studied stem cell niche, there's still so many new things to discover. And one of the things that our collaborators in the Fuchs lab discovered right before they reached out to start collaborating with us is a really, really close association between the lymphatics, which were until then considered just this sort of thing that absorbed nutrients, a very, very passive like pipes that ran through the system to do the collection and not active players in the niche. But one of the things that they really identified in the Fuchs lab is the really incredibly close and systematic proximity between the lymphatics in yellow and the really stem cells in purple here. And they really quantified that this is really remarkably close. And we see this both in mouse and in human, the same structure. So this is great. We can see the structure of these cell types, but that's only, 3 markers at a time. Wouldn't it be nice to build these structures genome wide? Before we went out to do that, we wanted to see, well, is this proximity? Does it matter? Sorry, I have a dry throat. And so we did some experiments to see what is the impact of lymphatics. First, in a co-culturing organoid system, and we saw that co-culturing with organoids really mattered. So organoids alone really grew these little mini guts with all these protrusions that you see in so many of these organoid gut papers. But when these were co-cultured with the lymphatic cells, these proliferated less and stayed rounder and more immature. They didn't develop, and basically the lymphatics were holding back the development of these gut organoids. This is in vitro. So we also looked in vivo to see do lacteals and lymphatics have a similar role. So we could actually do an experiment where we could ablate the lymphatics at a specific time and then see what happens. And what we can see is, first of all, and when we take away the lymphatics, when we ablate them, there are less stem cells compared to the control. And these cells don't die, but rather they begin to differentiate, as we can see, rapid proliferation under the ablated condition. Okay, so there's a huge functional role. So not only do we want to know, transcriptome-wide maps of what's happening, but we want to map the cell-cell interactions and the mechanism responsible for this tight control between lymphatics and the stem cells. But again, one of the problems when you're working with some of these beautiful protein-based, IF-based technologies is that you need to know what you're looking for. You need to know the player. You need to have antibody for it. You need to know what it might be. And it's sort of hard to guess who the player might be when you're in the situation where you can only look a few markers at a time. So this is where the wonders of spatial transcriptomics come in. There's a lot of different methods out there. What I'm going to talk about generalizes, even though we're using the Visium because it's plug, play, and easy. And we're not the first. In fact, there are many computational methods out there that what they do is basically take spatial transcriptomics, which gives us spatial resolution, but many individual cells, typically between 5 to 50 in an individual spot, and single cells where you do have full single cell data at the single cell level, but disassociated, and try to integrate it. Now, most methods out there, not all of them, are variations on the theme of some type of regression with correction, with the goal of taking each spot and identifying using the single cell data, what are the cell types that are present in each and every spot. And there are many methods that are pretty good at that. We decided that this is not enough for us. And moreover, some of these methods miserably failed for the rare cell types. And I really think that a lot of the most interesting biology is in the rare cell types. So we, the royal we being Tinny, developed Bayes prism. And Bayes prism is a Bayesian approach, which has many advantages, which I'll try to relate to you. First of all, we only use a single cell RNA-seq as a prior. So these are a prior on the expected cell types that we're expected to take.
Engineer.
And we don't take the cell type fractions into account, unlike other methods, because we actually think that in different regions of the tissue, in different individuals, these cell type fractions are likely to very much vary differently, and that could be confounding. So the single cell data, disassociated single cell data is the prior. The data itself that's going to really drive our inference is the Visium spot. And basically now we have for each spot an expression of what genes are being expressed in that spot. What Bayes prism does is it takes the prior and the data, and it basically learns a joint posterior. And we actually learn two components. We try and infer not only the cell type fractions in each spot, but we actually make it harder for ourselves. And for each cell type present, we learn the cell type specific count matrix for that cell type. So we have the cell type fractions, and for each of these cell types, the cell type specific count matrix. Now, this is a lot of inference for a lot of things, but actually we feel that the joint learning of these things together make each side more accurate in a way that best explains the data. And another thing is that these cell type specific fractions allow us to learn the variation of gene expression across different spatial regions. So this is why we only use the single cell data as a prior, but now we can see in our spot specific cell type specific count matrices some spatial trends in gene expression in a cell type specific manner. Of course, this is a lot of inference per spot, but because we're Bayesian, we can actually use transfer learning and share information across all the many spots across the Visium. So we do inference for each spot, specifically sharing a lot of the information across the spots, learning from the data rather than the prior. This is particularly useful for some of our cancer applications where each cancer can be very different from one another, and we could potentially learn a new cancer, which we haven't profiled in single cell RNA-seq using the Visium, because we can learn quite a bit of deviation from our prior. So taken together, what we have here is a spatially aware cell type specific gene expression matrix for each cell type and for each spot. Okay, so we can now apply it for our data. We collected single cell data enriched for both the stem cells and the lymphatics, which are very, very rare cell types. So we had to enrich for them. These are great in themselves. And now for each spot, we can identify the fractions. The first thing one does when they develop a new method is to try and benchmark it. The problem here with some of these benchmarking is it's very, very hard to get a gold standard for these methods. This has really been stumping the field. Well, we couldn't generate an extensive gold standard. We decided to generate a gold standard for what we really cared about. Since we cared about lymphatics and stem cells, these are the two cells that we want to sort of learn about and learn their interactions, we could stain these two cell types on the same Visium chip. So what we have here is IF profiling so that we could stain and identify in each spot, does that spot have a stem cell or not? And does that spot have a lymphatic cell or not? Creating a ground truth which we could test against. We could test both Bayes prism to see how well it does up here in the sort of the red up here. And you could see that we have a very good recovery rate, and compare it to all the other different algorithms. And you could see, first of all, a very, very high level of accuracy for Bayes prism with cell 2 location coming close behind, and some of the other algorithms doing less good. Again, this is a particularly hard test because I find that many algorithms, they look good at first glance, but rare cell types is the real test that really trips a lot of otherwise great algorithms. And some of the most interesting biology, I mean, stem cells, they're rare, is there. That's where I'm interested in looking. Now, remember, Bayes prism can not only identify cell type fractions, which I showed you that we did pretty good, but it can also identify cell type specific gene expression matrices. So we wanted to test how well we did here. We did 2 tests here. The idea of these tests is that we took 2 cell types that are known to be next to one another in the image. So these are cell types that tend to be next to one another. These are cell types that express the same gene, in this case, SOX9, but at different levels. And so here you see the fluorescence intensity. Here you see the normalized expression. And you could see that Bayes prism inferred correctly the difference in SOX9 between the stem cells and the Paneth cells that were right next to it. We did a validation like this in immunofluorescence. It also matched the measured FISH scores, and we did another pair of cell types showing that Bayes prism can really also get the cell type specific, spot specific count matrices for these cells, and that we could trust that. So we have spots, we have cell type fractions, we even have cell type specific count matrices, but how can we learn more? I mean, this is still very low resolution. We only have about 4 or 5 spots per crypt. How can we really learn interactions from that? This is really cutting the gut tube very rough. You could see these big gaps between each of these spots, another problem with the Visium. So here we decided to really use the canonical structure of the gut tube, and the fact that there's a very, very strong stereotypical repetitive structure of the gut tube. And we are in our Visium sampling many, many, many spots. And each of these spots fall at a slightly different point along this gut tube, but altogether it has a canonical structure. So we assume that the cell type fractions are a good representation of where we are along the gut tube. And we took each of these spots, used the cell type fractions as a vector, and mapped it down to 1 dimension, in this case using FATE, but most dimensionality reduction methods work. So now each and every spot is mapped onto a crypt-villus pseudospace. So we create a pseudospace and basically a continuous space to get really high spatial resolution for our many hundreds and thousands of spots, each one at a slightly different location in this pseudospace. And now we could look at the cell type frequencies as they change along this crypt-villi pseudospace. And for each individual gene and each cell type, we could see how that gene expression changes along the crypt-villus, giving us quite a bit of information which we can study. And again, pooling thousands of cells. First of all, we see that we really, really get the cell types distributions quite well. And specifically, if we're looking at the lymphatics, we see that some of the lymphatic spots are quite low there in the crypt, but some of the lymphatic spots go pretty high. These are the villus cells that are a bit different. And we want to know what's happening with the lymphatics in the crypt, the lymphatics that are talking with the stem cells. So we can ask what differs. So we're focusing on the lymphatics. We can ask, okay, what are all the main ligands that could be talking with the stem cells that are expressed at high levels in the lymphatics? And we found a few that are very specifically in the lymphatics. And one of these, relin, is specifically expressed at the base. So what we see here is as we go up the pseudospace, as we go up from the base to the villus, up the crypt, we see that relin expression peaks quite early on, right about where the stem cells are, and then goes down as we go up the villus. So we could validate using immunofluorescence that indeed relin is expressed exclusively in the crypt. And then we could do experiments, and we did both in vitro with our co-culturing system and in vivo, and really showed that relin, both in vivo and in vitro mimics the lymphatic ablation. So basically, whatever we saw with co-culturing with lymphatics or ablating lymphatics really phenocopied just by ablating or deleting relin or co-culturing with relin. So we both have activation and inhibition. So really, we learned that lymphatic, a new sort of player in the stem cell niche, are really relevant to stem cell maintenance. They basically control the stem state and sort of slow down the differentiation of these, finding a new player for a really well-studied systems. Now, while the proximity of the lymphatics and the stem cells were enabled by classic technologies, to really dig down into mechanism to find Rilin as a major player, a major ligand that's really driving this interaction, was really enabled by the computational methods we developed, Bayes prism and spacefold, which generalizes. So now again, data integration, because we want to go genome wide. I want to tell you another story of data integration. This is another algorithm by Rong. So a lot of people are trying to integrate not only Visium and single-cell RNA-seq, but single-cell RNA-seq and some of these fish-based technologies. Some fish gives you 30 markers. Some of these merfish technologies give you hundreds of markers, but the concept is the same. We want to map the RNA onto the transcriptomes, and there's, again, plenty of algorithms that do that. We want to go one step further, and we really believe in harnessing the cell's environment to learn more information. Now, what do I mean by environment? Well, each individual cell, sort of center anchor cell, has an environment, has a bunch of cells near it. And we could look at what's being expressed in each cell and each gene in its environment. This is its niche. Now, we're going to sum this niche as a gene-gene covariance matrix of the niche. So we're going to look for gene-gene covariance among the cells in each sort of center cell's niche, and we're going to compute covariance. Now, covariance is pretty heavy to compute. It's not the simplest and quickest of computations, and actually, I think it's a very powerful biological object that people haven't used as frequently as one could use because it is so heavy. And the one found a much quicker way to compute it. So now we actually can look at the niche covariance. So what you see here is a UMAP of the covariance, not of the expression, but of the covariance. And this tells you about the environment. It just tells you which cells are being are in each of these cells' environments. So each dot represents an individual cell. And the information we're plotting here is not that individual cell's expression, but what's going on in that environment. So again, to get this idea more clearly, the UMAPs that you're used to seeing, two cells are nearby on the UMAP if they are similar in their expression, in the expression of that cells, of two different cells. Here, we're actually not looking at the cell's gene expression, where two cells are nearby on this UMAP, on the covariance niche UMAP, if they reside in similar environments. So we're looking actually at the environment and not the cell. So we're going to use this sort of niche covariance as a driver for an autoencoder that's going to do this integration for us. So this is very, very similar to many other autoencoders that try and integrate these two sources, but we're going to do something similar. We're going to learn both the gene expression, the full transcriptome, but we're also going to learn the niche environment. We're also going to tell our autoencoder to learn not only the gene expression, but also the niche. And so again, we use this fast optimal transport distance to compute this in a way that can scale to millions. Because it's a conditional autoencoder, we could tell it what technology we're giving it, whether it's single cell RNA, merfish, fish, and we use different distributions to represent each of these technologies. And because it's a conditional distribution, we can get confidence for each of our inference, something that's not quite being used as often as I would like, but I'm a big believer in knowing not only your prediction, but how confident you are in this prediction. So first of all, when you integrate with NV, you see that whether you ran a single cell RNA-seq through the autoencoder or merfish through the autoencoder, you get a co-embedding that's very, very similar. This might seem obvious, but actually it's not. Most of the methods out there, if you actually look at the latent embedding and the labeling of the genes, the latent embedding doesn't co-embed the two technologies well. Now, this is another example where I could show why covariance, which is so hard to compute, is so important. Instead of using covariance, one could have said, why don't you use the expression in each niche? Why do I compute something so complex? I could just look at the average expression of the cells in each niche and use that. When we do the same thing, and we sort of run the same algorithm, but now we're just looking at the mean expression rather than the covariance of each niche, we see that the latent embedding doesn't co-embed. This is the first failure. Of course, everything else fails downstream of that. So it's really important to teach the autoencoder that in its latent embedding, it should preserve the covariance structure of the environment. So now we can see how good does it do in the task of imputing missing genes, everyone else does. Now, almost every method succeeds on neurobiology data. That data is really, really easy. It's really structured, and I find that test too easy. Therefore, we're going to use this sort of embryogenesis, gastrulation. This is from the mouse developing embryo, secfish out of the Marioni lab. And we can see that this is a much more complicated structure, so much harder to sort of learn and impute. And here we see on top the true values of these genes, and on the bottom, Envy's prediction, and you could see very high accuracy for very, very differing and complex structures that we see in the mouse gut tube. We can benchmark, and we can do it both on awesome fish and secfish, and show that Envy is systematically more accurate than some of the other widely used algorithms out there, such as Tangram and Jim's VI. Really, so the learning the environment helps also impute more accurately the gene expression, very similar to the situation with Bayes Prism, again, where we learned more to get even the simpler task more accurate. But in addition to learning the genome-wide expression and sort of mapping whole genomes onto the merfish, we could do it the other way around. We can learn the spatial environment and take disassociated single-cell RNA-seq data and see if we can guess where that cell came from, if we could look at the environment of that single cell. So going back to our sort of covariance UMAP, where each of these cells are mapped based on their environment, we could also map disassociated cells. So this is single-cell RNA-seq mapped to the environment that was imputed by Envy. So Envy thinks each of these single cells is placed into a very specific niche. The real advantage here is now we can look genome-wide, because in the single-cell data, each of these cells has the entire transcriptome measured, and we could look at lots of other genes that wasn't measured in the awesome fish. In this particular example, we see that GFAB is expressed at very, very high levels here, showing that it's really in a glia-rich environment as known. So really predicted or found known biology. Here, this is the merfish data. We can see that these activatory neurons, again, you can see how beautifully both the single-cell RNA-seq and the merfish co-embed. Activated neurons really map to activated sites in the data where there's a lot of an activated niche. Now, this is all 2D. And of course, if we want to learn atlases, we need to go to 3D. And again, as you sort of might have guessed from one of my earlier uses of gastrulation, embryogenesis and gastrulation is one of my favorite things. And so the goal would be to create a 4D map of gastrulation and really understand organogenesis from this. But of course, first step is to build a 3D map. So rather than building a 4D map, we want to build a 3D map of the gut embryo at first, and then see how the organs bud from it. This is a joint project with Katij and Tanakis, led by Mo in Kat's lab and Esther in mine. And again, this is really enabled by HCR, which gives you high resolution single molecule rendering of up to 5 molecules at a time. And you could really, I'm trying to zoom in to show you that the Cloudin really measures the gut tube, the white, and you could see different parts of the gut tube being demarcated by each of these. So now we could take each of our different color, different markers up to 5 and see at single cell resolution where each of these are being marked. But this is only 5 colors at a time, which we can map quite accurately. Now, what we'd really like to do is we'd really like to build a 3D map. And so the first thing to do is take different combinations of these 5 colors in 3D and align them together. And for this, I'm not going to go into all the details. We developed a method to extract from each of these gut tubes a common coordinate framework. So each pixel in each of these very different images, we can map to this banana-like coordinate framework, looking at the, and each dot knowing where it is on a mesh of an anterior, posterior, dorsal, ventral, and left-right axis. So now all our different gut tubes are sort of brought to a common coordinate. Then we could take all our different replicates, use our mapping algorithm to project them onto the common reference mesh. And now we could look at this common reference mesh and figure out an average expression for each of these, as well as a coefficient of variation to see, you know, what's very stable, what's very reliable, and what's not. We did this for 19 different markers. This is still work in progress. We're aiming to go to 40. 19 is not much, but again, we could use the same envy trick, our 19 reference genes and some single cell data that we collected from the same time point, and really try and map the entire genome 3D onto these 19 reference genes. Now, this is still very much work in progress. 19 isn't enough. We want to go to 40, but we're very happy. This is leave one out analysis. To the left, we see the atlas mean. To the right, we see Envy's prediction of this sort of leave one out, showing the pretty high accuracy we can get from this leave one out analysis. We can go back to our 2D map, our SecFish. So here we have more genes. We have a couple hundred genes in, I think about 400 in SecFish. And we could look at the sort of 2D slices and for markers that we didn't measure among the 19. So for new genes, we could see how well our data matches what we see in the SecFish, which is, again, isn't perfectly accurate in itself, but we could see very high concordance between our imputation of completely new genes that weren't measured with HCR and the SecFish data. And so we've developed this little tool that lets you look at about 2,000 genes, which we decided we have reasonable accuracy for. And again, this is going to greatly improve once we actually have more images to feed it with. This is really just a preliminary pilot, and you could look at each gene, what it's doing, and sort of look at its sort of 3D expression. Of course, the next thing would be to try and learn interesting patterns from these. But the first step would be to build a more accurate 3D atlas, which of course requires more data. So to summarize, new data requires new algorithms. We're still very, very early on, more working on data processing and data integration, trying to get some conclusions along the way. But there's a lot of algorithmic challenges. I hope some of these tools will be helpful for the community. And there's just so much more to do algorithmically. But new ideas, new ways to do autoencoders of moving pixels around, data integration through either Bayesian statistics or through autoencoders and deep learning, incorporating the environment and covariance as a major driver of learning are all things that I think are going to help the field. Finally, as always, we have both postdoc positions available as well as staff positions available at the Sloan Kettering Institute, and always are looking for faculty for next year's search already. And again, the wonderful people in my lab, particularly Doran, Tinny, and Esther, are wonderful collaborators. Elaine was a pleasure to collaborate with her dream team. Diksha, who's collecting amazing IBEX data, and Mo, who is a master of all HCRs in a long-term collaboration with Kat. And Lorenz is helping us interpret all this brain data. And thank you so much for your attention.
Thank you, Tana, for this fantastic presentation. A lot of useful tools for the community. Thank you for providing. And are there any questions from the participants? Please feel free to unmute. Alex?
Hi there. Thanks for a great talk. I had one question, one more technical, one more generalized. First, for the technical question, I was pretty surprised that for ENVI, the scRNA-seq and the spatial co-aligned so well. I would think that, necessarily for the RNA-seq, you'd be losing that context after the dissociation, right? So do you have thoughts for why it lines so perfectly? And then for the generalized question, this is more speaking as, not a computational person, but the landscape of techniques is really exploding. So how do people like us deal with all the new techniques out there, right? Like we kind of swear allegiance to one lab and follow their techniques, or what would you recommend for us?
Of course, swear allegiance to my lab and no one else. But no, I'll get to that second question in a moment. So that's the entire power of learning in joint embedding. And that's the whole idea of learning in joint. So when what we're doing in this, let me try and go back. One of the things that you do in this autoencoder and the whole power of this autoencoder is that we want this latent dimension, this L right here, which is trying to condense all the information and take all the information in a way that we can reconstruct from this L. So what autoencoder does is it takes the information that it takes, whether from single cell RNA-seq or from spatial, and it says, I'm going to chuck away all the data and only give you L. L is much lower dimension. L is much less. L is not the original data. And from this much lower dimensional L, what I'm going to ask you to do is reconstruct as accurately as possible the original data, both the gene expression and the environment. And we tell them, and what's unique about ENVI is we tell ENVI, you have to learn the environment. It's not enough to learn the gene expression, which is different than the other algorithms. So in whatever data you learn, you encode into it, in the whatever 30 OSM-fish genes or 155 MER-fish genes, it takes information from those 155 genes and says, I'm going to use this information to learn as much as I can about the environment. Then when we send the single cell RNA-seq data, there's an overlap between the 155 genes which it used to learn the environment and the 155 genome-wide genes measured in single cell RNA-seq. And that 155 genes, because the latent representation learned about the environment and the autoencoder insisted that it extract a latent representation that understands the environment when the cell is expressing those 155 genes, it is able to reconstruct the environment also for the single cell data when that didn't come. Because once you've sort of learned this latent encoding, it doesn't really matter when you learn it jointly if the data came from single cell RNA-seq or the data came from MER-fish. It has to, from only this latent embedding, learn both, give you back both the environment and the gene expression. So we're really telling the autoencoder, instructing it to learn exactly that, which is why it works. I mean, of course, there's a lot of tricks under the hood in order for it to actually work and succeed in learning. And again, Doron is incredibly talented. But first of all, we have to tell the autoencoder to do the right thing. And it's new that we're telling the autoencoder to learn the environment. As for what method, first of all, besides, what I just said, joked around earlier, there's no one best method. And you have to sort of really know what you're asking. A lot of methods are good for different things. Sometimes I develop multiple methods in my own lab that sort of do the same thing, but put stress on different things. For example, there are many methods that actually, as I said, fail in rare cell types. That's always the method that many of these benchmarks look at big averages of easy, broad cell types. I like to benchmark in the rare cells where most trip up. My advice is generally work with colleagues that know computation. It's always best to work in pairs. That's the most fun, and you learn from each other. Know what you want your algorithm to do. Know what you want your algorithm to ask. And even if you choose an algorithm, test it. Make sure that you get a robust answer. Make sure that even if you jiggle the data and try and trip it, you get the same answer. Really don't just take an algorithm off the shelf, blindly run it and believe it, even if you think it's good. But yeah, choosing an algorithm is not an easy task. That's part of the job of a researcher. And there's no right algorithm that's correct for all questions and all for data, period. Even the algorithms I develop.
Can I ask you a corollary about the first one then? How much information do you think how much spatial context do you think is lost in RNA-seq? Because I would imagine that, you have T cells that are in environment X or environment Y, they kind of lose that memory, some of it in the RNA.
Well, if you disassociate quick enough and on cold, so you don't lose that info, again, sample processing is, if you do poor sample processing, it's garbage in and garbage out. The idea is to process the sample so that at least the information is there if you have algorithms that can extract it. Otherwise, as I said, garbage in, garbage out.
And we have Path Kettering. You want to unmute yourself, Path?
Yeah, sorry about that. My computer just blanked for a second. I have two questions, I guess. The first is with Envy. Has there been any attempt to integrate with some of the single cell multi-omic technologies like CiteSeq or one of the combined RNA attack methods?
So, I didn't go into depth here because again, the Embryogenesis project is very early in its days, and I trust to only some degree our mapping based on only 19 genes in 3D in a process as complex as gastrulation. But actually, we did map multi-ohm attack in RNA onto the embryo and ran different things such as ChromeVar. And again, at a rough first degree, not nearly at the level of accuracy that I like to see. We actually now map not only RNA, but attack and chromatin onto our 3D structure. And if it looks that good with 19 genes, I'm really excited to see what the chromatin is going to look like once we have our full data set, which is going to triple the number of embryos we're using and extend from 9 and
yeah,
more.
that was kind of my question, I guess. Additional
there's no reason if you have the RNA there, there's no reason why, and the RNA is mapped well and correctly. There's no reason why you can't, bring any other modality that you want along with you. Here we brought the ATAC-seq along with us, and I'm super excited to see what it will allow us to learn about 3D configuration of ATAC-seq, about how the cells are setting up for embryogenesis. That's one of the goals. Again, I didn't go deep in there because this is still, a very preliminary project, and I'm showing you with like preliminary data that I myself personally surprised works that well.
Okay. The second question I had was also about the cell-cell communication. Do you think that since we are with spatial technologies moving more into kind of localized microenvironments, so to speak, does our understanding of cell-cell communication also have to change with potentially having a receiver cell receiving inputs from multiple possible sender cells, right? Because
I think we don't understand combinatorics, and most of our methods are linear, and certainly I think that a lot of signaling is not linear. And there's some nonlinear work. I mean, Mike Elowitz has some beautiful stuff. So I think now that we're accumulating data, we have to do two things. One, understand that cell signaling goes farther than one cell. There's way too many algorithms that look just at the immediate neighbor as if there's no diffusion. Yes, I think that too many of our algorithms are looking at single ligand receptor pairs rather than bigger swaths. But I think we're going to have a lot of surprises coming down the line now that this data is finally being collected. And one of the things that my lab is thinking of, and the answer is not trivial at all, is how do you model this in a more sophisticated way? And in part, what you said, cell is receiving multiple signals. How does it integrate it? Unlikely linearly.
All right. Well, thank you. The talk was fantastic.
Thank you.
Wonderful. And we have Hirak Sarkar.
Hi, amazing talk. Thanks a lot. So I had one question: is that what is your opinion about assays like SlideSeq, which has diffusion problem but gives a much bigger panel of genes? I mean, these are amazing methods, and it seems like when we couple them with single cell RNA-seq, that might reveal much more information about the architecture or so on.
So the entire main part of my talk, this one, it wasn't SlideSeq, it was Visium, which is the commercial version of SlideSeq. So this entire Bayes prism algorithm right here, and everything we discussed about our project was using Visium, which is a type of spatial transcriptomics, where SlideSeq is one of many, and there's a few others. Basically measuring genome-wide in these big spots where you lose a single cell resolution. And the whole idea of Bayes prism was to give you back the single cell resolution. And the reason that we succeeded in our project, and we succeed, like Relin is a gene that people didn't even think was expressed in the lymphatics. They actually thought it was expressed in another cell type. It was never shown to play any role in stem cells. Basically, if someone had to decide what gene to include in the panel, no one under the sun would have chosen this poorly studied, unrelated gene, which wasn't known to play any roles in stem cells, and which wasn't even known to be expressed in lymphatics. And the fact that we went with spatial transcriptomics and we opened it up to genome-wide, open-ended, unbiased exploration allowed us to find the surprising relin as a major factor that basically phenotypes the entire ablation of lymphatics. And the fact that we had such strong algorithms such as Bayes prism to do a very accurate cell type spot, geographically spatial deconvolution, and spacefold to really increase the resolution along the crypt allowed us to identify relin. Again, all these different methods, each of them serve a different need. And it's key, though, to have strong computation to analyze it. And this is not the end. This is just two algorithms along the way to try and get better.
The related question to that is that we saw that DEST-VI tries to do something very similar, mapping this transcriptional expression to a continuous space. So do you have any kind of feedback or observation when comparing to it or something like that?
So DEST-VI is the green. It's
okay.
Not terrible, but it's not great. Basically, in our hands, cell to location, which it's also Bayesian and has some conceptual similarities to Bayes prism, works really well. There's a bunch of recent publications really showcasing how great cell to location works. So if I'm to recommend algorithms, of course my own, but also cell to location, DEST-VI in our hands, also in other benchmarking that I'm not showing here, does not, it sort of conceptually sounds right, but when you actually benchmark it on real scenarios and not easy scenarios, it doesn't do that well, which is why I think that we're doing some of our benchmarking in these way too easy scenarios of, regression gets, neuronal structure right because it's so simple.
Yes.
And when you benchmark these algorithms in slightly harder context, I mean, like this one, I'm not showing, but the DEST-VI miserably fails here as well. It can't get the structure. So in our hands, DEST-VI has not been working that well.
Thank you so much.
And we have Hee-Sun Han.
Hello, thanks for the amazing talk. I really liked about the 3D work, especially because I do really think that 3D has to go when we're integrating all of these cellular interactions. But there, because currently the HCR can only do 5 at a time, you are averaging different tissue. So it's harder to look at like rare events, or what should I say, something that is not reproducibly probably happening with it, like across different tissues. It's harder to look at. So you're basically looking primarily at something that's more common, right? Like through the strip.
So you have to sort of choose the right problem as we go forward, and hopefully with time we will have stronger technologies. So first of all, I chose embryogenesis and this early gastrulation phase. And one of the things about gastrulation is this is one of the most robust and reproducible biologies. And so, yes, I'm choosing to look at an incredibly robust and an incredibly reproducible biology as a first shot. And I even started with the easy first phase, because you could also say, I'm aligning, I'm doing this alignment right here. And so you can think that this alignment isn't perfectly accurate either, which it's probably not. And so I'm starting with the simplest thing, which is a relatively smooth banana-like gut tube. I mean, it's not entirely smooth, but it's going to get much more complicated to sort of align these structures as organs begin to bud, and as you begin getting these lung bud structures. So I'm starting from the easiest thing possible, a very reproducible, very robust system, and a very easy geometric shape within the system. Next thing I'm going to go is I'm going to try and stick with reproducible and robust and begin looking at organ budding. That's when I get this first atlas done. And hopefully by the time I'm finished with that, there's going to be technologies that are going to give me a much higher plex. And then we could do whatever we want, tumors growing in all sorts of weird and non-canonical ways.
Thank you.
Wonderful. And we have a few questions on the chat box. So Benshia is asking, does Bayes prism detect expression or location of transcribed cis regulatory elements, such as enhancer and transposable element?
Where is it? A bit,
That's on the chat box, the first question.
yeah. So again, right now, Bayes prism is just using the standard count matrix as input. And also, that's what you get. One can imagine that if you build your count matrices, both on the Visium side, I'm not sure if Visium is able to capture these well, and on the single cell side, then you can have count matrices with more element, because it's how you process your count matrices and what you include in your count matrix. Now, Bayes prism, and we've benchmarked Bayes prism extensively, works very, very well when you have a bunch of unique markers that really define a cell type. So if you have a cell type that's really defined by a set of markers, for example, a T cell with CD45, CD3, and a bunch of others, and you could say, okay, here's a marker that really identify this cell type correctly, then as long as you sort of have that going for you, you can add a lot of other elements, including noisy elements, into your count matrix and actually reconstruct them pretty well. But you'd have to go back, chuck Cell Ranger, go back to the raw data and build the count matrices, including whatever elements you want to include there, which can be dicey. Going back to the raw data, you need to sort of know what you're doing. There are many artifacts there.
And we have also a second question from Hira. Can you see the chat screen now, or should I help you?
That's already answered. I'm sorry, I asked twice.
Okay,
That's already answered.
wonderful.
Oh, when will NV be published? When Doron writes his, he's probably listening right now. It will be published when he finally writes it. We'll put it up on bioRxiv the moment there's something that can be shown to the public, but it's in drafts right now, and my goal is to get it out to the community as soon as possible, hopefully within months.
Wonderful. So are there any other questions? Please feel free to unmute or post on the chat. In the meantime, I have one question, Dana, for you. The new map you showed, right, where you mentioned that some part of it, when they're close, that there's proximity in space too, right? That's, your expectation. So I'm kind of curious about, for instance, if there are two cell types, right, that are similar expressions, right? Is it the expression leading it or the position leading it? Could you elaborate on this a little bit for my understanding?
Yeah, so again, trying to go back here. What we're doing usually here is here we're actually mapping the latent embedding. And the latent embedding is often driven, as you can see, more by cell type than by environment, because this is sort of the embedding that embeds both the gene expression and the environment. Here, for example, when I'm showing you this right here, I'm taking these purple cells right here from latent embedding and creating a new embedding, which is only the inhibitory neurons. And for those, I'm mapping them based on their covariance rather than their gene expression. So only in this UMAP, which we typically do on a cell type specific way and all cell types together, though it could be interesting to do that, do we have that spatial proximity. If there's this algorithm NovaSpark that was published by Mor-Nitzan, and yes, in NovaSpark basically uses the idea that nearby genes are going to have more, nearby cells are going to have more similar expression. And NovaSpark alone, based on that simple assumption of if you have more similar expression, you're more likely to be nearby, allows her to do, actually NovaSpark works incredibly well for such a simple assumption. So that tells us that spatial localization has a very, very big impact. But of course, there are many other factors. For example, what cell type are you? If you have a T cell next to a macrophage, they're going to be very, very different from one another. So basically, it's very interesting for me, and this is something that I'm, again, on the long list of things I want to do that are endless, really want to take the gene expression and deconvolve it to its different impacts. What gene expression is driven by the past history of the cell? What gene expression is driven by the sort of the fate cell type determination of the cell? What gene expression is determined by its environment? And sort of really what gene expression is determined by some cyclic things like cell cycle, and really break it down to these multiple influences. They probably have different structures, so it's probably something that can be found, but space definitely has a big impact on expression.
Exactly, and also the nonlinearity of these maps, right? And how the space is more, I think, linear, right? As you move you move with the speed, and right, there are physical rules. So how are they being matched?
So first of all, 3D space is certainly, if you're trying to squish 3D space into 2D, it's nonlinear. So, cells are maybe moving in space, and maybe some, like, for example, if you think of a tumor niche, we see a lot of different gradients of hypoxia and of glucose and all sorts of other goodies. So while any individual stimuli might be linear, because we were earlier discussing that we think actually the way the cell integrates this information, and there's definitely been some work both by Elowitz and Alon and some others that show that a lot of the signaling processing is nonlinear. So what I think is happening is that a cell is maybe taking some linear gradients and creating sort of nonlinear, just nonlinear output from them. And also some of the nonlinearity, especially when you have these cells each doing that and interacting with each other. We see, for example, these little tiny circuits that were shown, particularly in immunology, particularly between immune cells and fibroblasts, where you can get this little circuit of a bunch of cells that interact with each other and take that little mini niche into a completely sort of new state, which within that mini niche is very different than, as you sort of zoom out. So there's a lot of mechanisms, feedback loops, combinatorial integration, that can create a non-linearity of behavior within large linear gradients in space.
Wonderful. So therefore, we expect that actually the nonlinearity can be mapped on nonlinearity nicely.
A cell's circuitry is very complex. We aren't anywhere near to figure it out, and anything that goes from the linear to the nonlinear, most of our modeling is still very poor.
Exactly. And as we are waiting, by the way, I'm monitoring the chat and waiting for someone else to also ask. But as they are waiting, I'd like to also ask your vision to implement RNA velocity type of analysis in spatial community, right? Because you look at these variances, and each variance could be like RNA velocity, right? Those splice versus unsplice and whatnot, and that's time dependent. Do you have any vision for where the field is leading with the spatial?
So actually, I didn't show it here, but Tinney ran velocity on our sort of spatial rendering of the gut tube, and it actually worked quite well. So once we sort of had the Visium and we mapped cell type and gene expression, to each thing, we sort of ran velocity in that, and that worked reasonably well, surprisingly well. Again, as long as you can get the data spatially mapped at high accuracy, there's no reason why you can't then pull down all the other algorithms that we've learned to do quite well in the single cell space and map it spatially. That's the whole power of these sort of integrative approaches that map big single cell data sets into a spatial context.
I see. And also, not just RNA, can we think about velocity in protein context too, right? Like, for instance, developmental axis, right? Can we map it using proteins and things like that?
So again, many of the protein technologies, certainly protein. So we're really behind in the protein technologies. Much protein regulation is based on phosphoprotein, which is seen less. One of the things that we've been developing in my lab is RNA based on ataxia, which we call regulatory velocity, where again, if you can understand the regulatory network and the open regions that are available for regulation, similar to a velocity vector where you can guess where a cell goes, you could use ataxia to sort of guess where a cell goes. And that I think will work very well in these spatial methods.
Wonderful. Yeah, we played with ATAC-C from Howard Cheng's lab in the past. And it's interesting to see the dynamics in 3D in chromatin as well. And are there any questions? If not, then let's thank to the speaker again. And I'm gonna use this clapping sign as well as thank you, Dana. I think this was a great presentation. And with this, I think today we will be wrapping up. But before we do that, I'd like to also announce that next week, we will be hosting Dr. Amjad Askari, actually the postdoc from Ilowitz's lab, and now he's assistant professor at UCLA. And he's gonna talk about imaging-based genetic recording of developmental history. And that's a technique called, I think, Zombie, published in Nature Biotech a couple of years back. So with this, we're going to be wrapping up today. And thank you again, Dr. Pierre, for this opportunity to get to know you and your work.
Okay, thank you everyone for listening, and see you at conferences in the future, now that we're going back to a real world.
Yes, very exciting times. And
Yeah.
have a good weekend, and see you guys all next week. Bye-bye.
Bye.